So we're talking about mitochondria. Today we'll be talking about mitochondria. From childhood, I say we've been studying mitochondria. It's the powerhouse of the cell. We need to increase that understanding to a small extent, I guess, and we need to understand that mitochondria is the site of ATP synthesis, and ATP is actually cell currency, or simple understanding the energy of the cell. Okay, and why do we need energy? To do everyday tasks, to generate heat in our body, to produce heat in our body, okay, whatever it is, and for various works. And so distribution, we'll be talking about distribution of mitochondria in this video. So mitochondria, distribution of mitochondria can be explained in two major ways. That is simple understanding. So how many cristae are present in the mitochondria? So it's simple understanding, cristae are enfolding. So mitochondria is double membrane system. Okay, mitochondria is itself is a double membrane system. I'll write over here double membrane organelle, and I know every one of us knows this. And the inner membrane has foldings called cristae. Okay, these cristae, if on the basis of presence of number of cristae, we can divide the distribution of mitochondria in the cell. Okay, so if there is many cristae present in the mitochondria, it could be related or associated with mechanical work or osmotic work, these type of work situations. So if it is mechanical work situation, we'll be talking about osmotic work situation in a while. Now talking about mechanical work situation, those cells of the body that require a lot of energy to perform a lot of tasks, that is those have many cristae in them. So whichever cells will have a lot of work, that is muscle fibers, then the basal infolding of kidney tubule cells. Okay, not only kidney but basal infoldings, and the inner segment of rod and cone cells of retina, and many more. Okay, so they require a lot of cristae, presence of a lot of cristae for a lot of ATP synthesis to do a lot of work. So talking about a specific kind of muscle fibers, sarcomeres, they have the myocardial muscles of the cell, muscle cell, that is the muscle cells of the heart, have many mitochondria, because they have to keep beating a lot, all around the day. Okay, and so I said, so they can be found in so second way of classifying uh, mitochondria is presence of mitochondria in a congested, being associated, presence of many mitochondria in a cell, presence of many mitochondria being associated at one particular region or one particular corner of the cell, or being evenly distributed, like in the cells of liver cell, like in the liver cells, mitochondria are found spread throughout the cell. This is actually because the hepatic cells are mainly related to biosynthetic biosynthetic and degradative works, degradative work. That means they are for photosynthesizing and degrading things. That means they need efficiently large amount of mitochondria all around the cell, distributed all around the cells. Whereas like in the cells oocytes of a specific marine animal, thion, Briaeus, thion brius, it can be found many mitochondria. I'll just do like this. Many mitochondria are found being associated with RER. Okay, being associated with RER, and so see, it can be either found uh, at a particular region, or it can be found being throughout the cells, spread throughout the cells. It is not spread, it is spread. Okay, now we talked about mitochondria having many cristae for mechanical works. Next work is active transport work. Simple understanding, active transport is those type of transport of certain things, generally protons and sometimes electrolytes like sodium and potassium, in the presence of ATP, in the presence of energy. So in those type of cells where osmotic works occur with active transport, those type of cells also need a lot of ATP, because ATP is required for the transport, and that is in those cells many cristae are found in order to synthesize many what do you call many ATPs. So I think I did not write where can it be found. So I'll write in very small over here. These can be actually found in the cross border of kidney. Okay, you can say brush border of kidney, in actually in proximal tubules, and in the infolding, and in the malpighian tubules of the insects, and many other things. But this one has to be understood, right? Brush border of the kidney. Now let's talk about cells needing less cristae. So not all cells need a lot of energy. They may need very less energy just to thrive, just to live, and those type of cells will have lesser cristae. Those type of cells will have mitochondria with lesser cristae. And one particular striking example is non-myelinated axons. They are very less. Most of the case, only one cristae is present in non-myelinated axons, but still they may have many mitochondria. Okay, so the many mitochondria is not associated with a synthesis of ATP. We know ATP synthesis won't occur much because of the presence of only one cristae, but they have many mitochondria because they have an enzyme named monoamine oxidase, or MAO, which is actually found in the outer membrane of mitochondria. This actually helps in breakdown of monoamines. So what is monoamines? So simple understanding, monoamines, breakdown of monoamines are neurotransmitters in the axons, and we know neurotransmitters are required in the neurons. And deamination means simply breaking down of monoamines. And so example, dopamine, or norepinephrine, and serotonin. And why is the breakdown of monoamines are important is, it is that if there is excess monoamines present in a non-myelinated axon, then that can get accumulated at certain portions, and that becomes toxic to the cell. So in order to prevent that accumulation of the monoamines, MAO, or simple understanding, monoamine oxidase, is required, and for that mitochondria is present in a large number. So this is all about distribution, or simply understanding localization of mitochondria. Everything I taught is actually from a book called Cell Biology, Genetics, Molecular Biology, Evolution and Ecology by Pierce Verma and AGARWAL and Vicky AGARWAL. Okay, if you want to go through this book. Okay, this much for this video.